Today we're going to start our classification unit. We are going to start by discussing trying to find some order for all of the diversity that we have in our world. So what we're really talking about is taxonomy, and taxonomy is the science of naming or classifying organisms. So it's classification, and it is based on the idea that everything in our world is related in some way. So all living things have some type of connection. And the way scientists group and classify things is based on their structure and function. What scientists have also done is they have also assigned scientific names. Common names are like what we call animals. Scientific names are based in Latin, and it's what scientists use while they're discussing organisms so that there's no confusion as to what they're talking about. A good example for you would be, the little bugs that you chase around in the summertime that their butts light up at night. Some people call them fireflies, some people call them lightning bugs. Because common names like that can vary from place to place, it can sometimes get confusing as to what animal you're talking about. So another good example of that is the animal cougar, puma, panther, and mountain lion. They're all actually the same animal, but what they are called in different regions around the country is different. Their scientific name is actually Felis concolor. Binomial nomenclature. So in the 1730s, Carl Linnaeus is the one who designed our naming system, our scientific naming system called binomial nomenclature. And that is what our scientific names are. Our scientific names are all based in Latin. What Carl Linnaeus came up with was the first scientific naming or classifying system, the first formal system of taxonomy. With what he came up with, the first word is always a capital letter and the rest are lowercase. The first part of the word is the genus, and the second part of the word is the species. So Homo sapien, that's the scientific name for humans. When we look at Homo, that is the genus, sapiens is the species. Typically, though, when we say the species, we say Homo sapien and put both words together. In addition to naming and organizing things, biologists always try to organize things that were once living into larger groups in order to help give them meaning. Your larger groups from your classification is a taxa or taxon. Linnaeus developed a classifying species that was organized into a hierarchy. We've done things with hierarchies in the past. In deciding how to place an organism into a larger group, he based them and classified them based on their anatomical similarities and differences. Your taxonomical classifications used by our scientists are a part of that nesting system, where you think of like your Russian nesting dolls, where you open that doll, that little wooden doll, and then inside is a smaller one and a smaller one till you get down to the teeniest, tiniest. That's what this is. We start at a very broad classification level, which is our domain, and we go down into a very specific one. Each of these groups is called a taxa. So we're going to work from the smallest to the largest. Species is the most specific that you can get. A species is one that can breed with each other to produce a fertile offspring. Genus is a group of species that are closely related and share a common ancestor. Above genus is a family, and it is a bunch of related genera or the genus, excuse me. From there, we go order, which is a bunch of related families, class, which is a bunch of related orders, phylum or division, which is a bunch of related classes, kingdom, which is a bunch of phyla, a domain, which is the kingdoms, and there are 6 kingdoms. That ends section 1. We're going to talk about section 2, modern evolutionary classification. So when we are talking about evolutionary classifications, this is grouping species into the taxa based on a common ancestor. Phylogeny is the study of living and extinct organisms and how they are related to each other. A phylogenetic system is grouping species into larger categories that reflect the lines of evolutionary descent rather than the overall similarities and differences. A clade is a group of species that includes a single common ancestor and all descendants of that ancestor living or extinct. A clade must be monophylogenic group. This means that all the species are descended from a common ancestor, and they cannot include any species that are not from a common ancestor. Your cladograms are a method of determining how clades are related to each other. This information is used to link the clades together into an illustration called the cladogram. And it shows how evolutionary lines can branch off from common ancestors. What you see down there is you see the building of a cladogram. And what we have is a branch into the different lineages from that branching point or node. The node represents the last point at which the new lineages shared a common ancestor. So when you're looking at a cladogram, the patterns are going to indicate how closely related they are. So when you're looking at 3 and 4, they share a common ancestor more recently than what you see in 2. And 3 and 4 are more closely related to each other than they are with the other lines. Cladistic analysis also focuses on certain characteristics called derived characteristics. A derived characteristic is a trait that arose in the most recent ancestor and was passed along to the descendants. Derived characteristics are present in the members of one group of the line, but not the common ancestor. So it's the more recent ones and not the ancestor. When you're reading the cladogram, it shows the phylogeny of the cat family. So what we're looking is how closely related the cats are to these other animals. So at some point, these animals shared a common ancestor, and the further you go down to a cat, the closer related they are. So when you look at this, this shows the order in which those characteristics arose and how they evolved. You can use genes as a derived character because we know that DNA is passed from one generation to the next generation. Because genes mutate and change over time, we can use this as a derived characteristic in analyzing our cladograms. For that reason, similarities and differences in DNAs are used to help develop evolutionary relationships. So we have a tie between classification and studying evolution. Our new techniques using DNA in this form of analysis are helping us to develop new trees and fixing what we thought before. An example of this is the American vultures and the African vultures. They were once classified together in the falcon family, but when we looked at their DNA, we actually were able to see that the DNA from the American vultures is more similar to storks than it is to the DNA of African vultures. So then you would have to go back and redo those cladograms to reflect this. Scientists are able to use this even when we are looking at anatomical traits. Sometimes we look at the anatomy and they look like they might be related. An example of this would be the red pandas, which you see a picture there on the left, and the giant pandas. They share some characteristics with bears and raccoons. They had some things in common. But when we did DNA, what we came out and found was that your giant pandas have a more recent ancestor with bears than with raccoons. So we would place the giant panda in a clade with bears. The red pandas are actually in a clade with raccoons and other animals like weasels and seals.